All right, so welcome everyone to the AESP Student and Postdoc Services Committee Future Faculty Seminar Series. This is the first seminar of 2025. We have two excellent speakers today. And now I will turn it over to our co-hosts, Sudeep and, I'm Jessica Wilson, by the way. Sorry, I should introduce myself. I'm the chair of the Student and Postdoc Services Committee. So again, welcome. And so I'll turn it over to Sudeep and Fab, who will be the co-hosts and who will be introducing our speakers. All right, thank you.
Hi. Thank you all for joining. It is my pleasure to introduce the first speaker for today, Hannah Spiro, who is a PhD candidate and an NSF Graduate Research Fellow in the Department of Civil and Environmental Engineering and Earth Sciences at the University of Notre Dame. Her expertise is in the area of environmental fluid dynamics, and her research spans extreme wave events and integrates numerical modeling, field campaigns, and geomorphic analysis to advance the understanding of community resilience among those geohazards. And so with that, Hannah, take it over.
Wonderful. Well, welcome everyone. I'm excited to share a bit more about my research broadly and then my doctoral research more specifically today in a talk titled When Storm Waves Move Boulders, Linking the Field to Models. A bit about my background, as was mentioned, I'm interested in extreme wave events. So subduction zone tsunamis, which we see here on the top, catastrophic dam failures, landslide generated tsunamis. And then what was the focus of my doctoral research was on storms, and not just storms, but also their geomorphic evidence. How do we build the record of past events? So with that, what are our motivations for researching storms on coasts? There are increasing coastal populations, not just on sandy coasts, but also on rocky coasts. Rocky coasts are estimated as over 60% of global coastlines. There's climate change-induced sea level rise, the storm surge, inundation, and flooding. And with the interaction of all of these components, there's an urgency to protect coastal populations and infrastructure. Specific to the island I work on, Inishmaan in Ireland, that we'll be traveling to today, they're wholly dependent on cargo access for food. And so there's an urgency to studying these hazards. On rocky coastlines, they're highly energetic. So often the only way to build back this record of previous events is using coastal boulder deposits. So you'll see a coastal boulder deposit here. This is on the island of Inishmaan that was formed during one of the storms during our field campaigns. And these boulders are above the tidal range, so they're not accessed by everyday workaday waves. They're only moving either in storms or in tsunami events. But that's part of, as you'll see, the issue is differentiating storms versus tsunamis. And that comes down to how do we link local and regional wave climates to the transport of these boulders and the formation of these deposits. And they're useful in coastal hazard management, think inundation or flood mapping, evacuation mapping, and then also recurrence intervals. And some of the gaps in knowledge that exist in this field that I'm working towards include the lack of representative wave heights to characterize these boulder deposits. So these boulder deposits exist all over the world on rocky coastlines. They're most researched, as you can see here, in Europe and in Japan. But if we don't know the wave climate, then we can't interpret if the boulders are from storms or tsunamis. And then what further complicates interpretation are site-specific methodologies. So there's no yet field-accepted standards. So if we're not standardizing the way we calculate volume or that would quantify the minimum wave velocity needed to move boulders, it's difficult to have a generalizable framework to interpret these models. And there's this quote from one of my collaborators, Rona Cox, that I really like, where she states that the consequences of misinterpretations are substantial. So not only does it undermine forward planning with respect to coastal hazard management, but also potentially public trust in science. And in this day and age, public trust in science is critical. So with that, where did my work come in? So I've been working for the past 4 years on a project where I led on-the-ground efforts. This is my Bitmoji. I led on-the-ground efforts in Ireland on an island called Inishmaan, piloting new methods to collect data on when boulders are moving and linking them to waves. I am co-advised by Dr. Johannes Westerink here at Notre Dame. This first part was under Dr. Andrew Kennedy, but I worked with Dr. Johannes Westerink on using regional wave modeling outputs so we can contextualize how these boulders are moving. And then we have 2 interpretive and generalizable frameworks that we're using for these boulders. So we're traveling to Ireland today. We're on the west coast here in Galway Bay on the island of Inishmaan, which is in red. You'll see a nearby tidal gauge that is in yellow. And Inishmaan has been studied in terms of large mega class transport, so over 600 tons, boulders that are being moved by storms. But folks have not looked yet at annual level effects. What are the preservation potential implications for these deposits? Can common storms move boulders? Are they moving more frequently? Which is part of our study. There's also active clifftop deposits, so over 30 meters above mean sea level in height. And you can see an example of a storm wave that was from Graham Brutton on TikTok, where it's overtopping those cliffs. So we had a lot of different field methods that we were testing and using in tangent, kind of like detectives, to figure out what exactly was occurring on the coast. So before each winter, so for 3 winters in a row, I moved out new boulders from the ridge towards the sea, where they would be in more vulnerable positions to be moved if a storm wave reached that level. Then we would drill and install accelerometers. And in the second year, I pilot tested Apple AirTags inside boulders to see their changes in orientation. And then Apple AirTags are useful because we have to retrieve the accelerometers at the end of winter. So we had to be able to find the boulders. And you'll see an example here to the left of the Apple AirTag screen. I'm a drone pilot, so we flew surveys, uncrewed aerial surveys, where those would provide timestamps of centimeter scale resolution where the boulders were at. And we were able to process those using structure from motion into digital surface models. And you can tie those DSMs to Earth using real-time kinematic, or these RTK corrections. So you can get the elevation, the latitude and longitude of all these locations. There are also the traditional field survey methods. So taking notes, the A, B, and C, or the longest, intermediate, and shortest axis measurements of the boulders. So you'll see I taught a field school, and these are 2 of the students measuring one of the boulders, and also the handheld GPS locations. 2 more methods that I wanted to highlight were in year number 2, we had pressure sensors that we installed near some of the study boulders. So far, I had talked about characterizing the setting and characterizing the boulder. The pressure sensors allow us to have onshore inundation measurements at the time of movement, which is extremely useful. So altogether, the 4 sites I'll talk about today, Site 1, Site 2 on the southern coast, Site 4 is a clifftop site, and then Site 3 here in the west were studied in depth with boulders spread throughout. 50 boulders total in year number 3. We moved boulders, as I mentioned, from the ridge towards the sea, and I have 3 pop-out method slides on the methods. And that's because one of the things I believe I'll be known for coming out of my PhD is not just the results, but also how are we coming up with new and innovative methods to study these complex landscapes. So one way was not just winch moving these boulders, as you can see here, but having the idea to install a dynamometer. Because if we have, for example, assuming a slope of 0, we'll be able to calculate a coefficient of static friction if we could simply quantify that pull force. And this coefficient of static friction has been thought to be important in those interpretive equations I mentioned earlier, but has not previously been quantified in the field. So this is allowing us to have a first order quantification of that parameter. I mentioned earlier the accelerometers and Apple AirTags. So accelerometers had been piloted before in a Stephenson and Ava Zovic study, but for a 3-hour short study and a smaller class. So we scaled up that method for 150 days for 3 years, so covering 3 winters, and sampling at the rate of every 10 minutes. So every 10 minutes, we can calculate the change in orientation from its initial orientation. So we're getting if boulders are sliding, if they're rolling, and these associated wave energies with those movements. And another method that I'm really excited that came out of our work was I had the idea to use iPhone LiDAR to scan these boulders in the field. We only can work when it's low tide, when there's no storms, when it's not slick. And so those conditions can inhibit, even when you can get over to Ireland, your opportunities to look at these class. So why not bring them back to a virtual laboratory that you can have, for example, here at Notre Dame? So you can scan these class using iPhone LiDAR, and then I came up with a method to post-process those using Blender and open source software into 3D watertight models, such as that you see here on the right. And using a 3D print tool, you can simply calculate the volume. You can also orient it to calculate drag areas and lift areas. And we compared this with the A, B, and C, or the cuboid assumption of the boulder volumes, and found all of our complex volumes or 3D volumes were smaller as a method of validation. So that's an exciting method that came from my PhD. So now I'm going to move on to results. I'll spend most of my time discussing 3-year field campaign findings. I'll spend some of my time contextualizing the boulder transport results with the local wave climatology, so onshore depths, minimum flow velocities, and then I'll give a teaser of the 3rd chapter of my doctoral research, which is using the numerical modeling results to link offshore to onshore. So to begin with, in literature, it was largely assumed the 3 most important first order parameters determining whether or not boulders will move are elevation, seaward extent distance, so from the boulder to the high tide line, and then microtopography, which here we can think of as there could be obstacles or no obstacles that are governing whether or not a boulder will move. Winter 2022-23 is year number 1. We moved a lot of boulders, most boulders in our study, we moved using that winch method towards the ocean. Once we moved them, we left them longitudinally in our study, but we did not move them again. We'll notice here at Site 1, which is in blue, there's much smaller boulders. Most of these boulders are moving. Most of these movements all occurred in one storm event, in November 2022. So these boulders are moving, and then we compare that with year number 2, for example. We add a clifftop site, so that's our site number 4 in year 2, and we see elevation as a key control, at least in this case, so ranging from 19 up to 27 meters in elevation above mean sea level. The storms were considered annual level or common this year. They did not reach these boulders. Whereas if we look back here at the blue boulders, they moved during year number 1. Most of them moved back into the ridge, which we would consider an obstacle or the influence of microtopography, and they were considered stable, so they did not move in the 2nd year. We can look from a top-down view. So this is our process digital surface model of a boulder deposit at Site 3. We'll see the 3 boulders here, boulder 2021 and 2022. And in white is their pre-winter position, and in red is their post-winter position. And to the north of the screen that you're seeing is the seaward distance. So we can look at not just where boulders are moving, but sometimes when they move, they leave scratches on the pavement, on the limestone pavement, called abrasion trails. So we can use those to recreate if they're moving along a ridge or in straight linear distances in these pathways. But not only can we look at that, we can look at when they're changing in orientation, which is key. That's something that this study is contributing to in terms of literature. So we have an initial position and a final position, and we're just calculating that angle of difference between those two. So for example, Boulder 21 and Boulder 22 are both moving in this November storm. They're moving in different ways. So a 60-degree orientation change would still be considered rolling of sorts in our broad categories, but a 180-degree orientation change is indicative of a much higher wave energy event that's changing this boulder's orientation. Boulder 20 is stable in orientation for this first year. So we were excited about our results from NERO number 1. So we moved out 6 new boulders here. And as I'm explaining this, I'll also be plugging in my computer. But we had Boulder 28, which you'll see here on the left top of the screen, which moved 33 meters. So this was quite a far distance. It's the largest at this site. At other sites, we had boulders that were moving up to 90 meters, which is substantial. But we found that storms coinciding with high tides were almost always necessary for transport in the city, which is an interesting finding because traditional models neglect the inclusion of the tidal range when interpreting these coastal boulder deposits. Additionally, we were interested, since this is year number 2, in looking at the pressure sensors, which are these 2 red dots that are here on the seaward side of the platform. We were interested in looking at what those were logging. Were all of our accelerometer changes in orientation associated with high tide, associated with what kind of inundation? So we can first look at, and these are masses computed using a density 2,610 tons per meter cubed, but using the volumes, the 3D volumes that we came up with. And then we can look at those first for a first order mass threshold that we find. So the much larger boulders did still move, but moved by sliding. Smaller boulders, perhaps roundness or sphericity could be linked to how they're moving. So that's actively under investigation. One boulder, Boulder 26, actually jumped. It ended in a position like a snowman as the top boulder. So we can look and then dive into these accelerometer records. So for example, we have that Boulder 26, where in late January, there was a Storm Isha, and that was associated with that snowman position at the end. Boulder 23, which we can see is one of the smaller boulders at this site, moved quite far, moved against a ridge, and substantially changed in orientation. So in year number 1, we found that boulders were moving, coinciding almost exclusively at or near high tide. And this is high tide that's both spring high tide, also neap high tide. Just changing that instantaneous water level so it's easier for the storms to reach these deposits. And we can see here the M3 and M4 Irish wave buoys. So these are over 200 kilometers away from the site, but are the nearest wave buoys with open source data. And we're seeing over 7 meters significant wave height in some of their cases, and the storms coinciding with high tide, and these boulders are moving. So in year number 2, we have the pressure sensors. So we're able to look at onshore inundation. So you'll see pressure is on the y-axis, time, and this is just 1 day, is on the x-axis. And we'll see all of our accelerometer timing events are coinciding with measured inundation onshore, which is a pro for the method. Our boulders are not moving or not changing orientation with respect to any other forcing. And then we can look and compare the different pressures at the different sites, the different levels of movement. Now, pressure is useful, but it would be more useful to convert it to depth. So I came up with a method where during storm intervals, which you can see here in this first plot, we took the absolute pressure, which is what our pressure sensors are logging. So we can see that here in orange. And then to use the hydrostatic pressure equation to convert this to depth, we needed to calculate an atmospheric pressure. So we determined the floor during these inundation peaks. And then in special cases, when we had the accelerometers changing in orientation within 10 minutes, we could use the 10-minute maximum pressure. So what that actually looks like is, so for example, here, Boulder 26 had 3 back-to-back movements. So we have those 10-minute intervals. And we're able to calculate, and this is compared with the height of the boulder or the c-axis, whether or not a boulder was submerged when it was moving. And this is important because traditional models assume full submergence of these boulders. But what we're finding in our results are over 60% of movements in our study occurred when boulders were partially submerged. And partial submergence does not necessarily indicate one would slide or one would roll. Boulders are moving by both mechanisms, which is an interesting finding. So that prompted me to revise a model from Nandasina et al., where we have, it's a force balance model, and we're wanting to determine the minimum flow velocity needed to initiate transport. And we could calculate this, excuse me, for sliding, for rolling, or saltation of these boulders, considering full submergence. And then I worked to recalculate drag areas and volumes for boulders that were partially submerged based on those depth estimations. And we were able to compare those two. And what we found is when a boulder is partially submerged, the velocity needed to move that boulder is higher, which makes sense based on the buoyancy force and also this reduction in drag area. But it's an important finding because if we're interpreting boulders where we don't know their wave history, we need to take into account a range and a potential that it could move when partially submerged. And this aligns with findings from Ressin et al., a lab study, and then one field study with video evidence. But it also has implications for, and this is one boulder, an example, with a 10% reduction in drag area, partially submerged volume, and looking at the effects on velocity and the Froude number. But we're finding the apparent Froude number increases almost twofolds when the boulder is continuously partially submerged and reduced. And we can look also at the coefficient of static frictions that we were measuring using the dynamometer. And that has a 1 to 3 meter per second order of influence on our boulders. But most folks in this field studied the largest boulders at each site. And in those cases, it's critical to measure the static friction because it's increasingly important as you increase the mass. And one other note is following up with the apparent Froude numbers. So using that minimum flow velocity we calculate, and then here the onshore depth as the H value or the representative length value, we can compare the commonly assumed Froude number of unity for storm transported boulders with what we measured. And we're finding our apparent Froude numbers are on average much larger and are falling more within the range of tsunamis. And I promised a teaser, so I'm actively working with an AdCirc SWAN output package that was ran by a lab colleague, Amand, and my co-advisor, Johannes Westerink, where we have the whole domain of the Atlantic, and we have wave periods, offshore significant wave heights, and we can combine those into this framework where we're interpreting coastal boulder transport onshore and also linking offshore wave directions with boulder transport directions. So the goal of this work was to develop linkages from offshore to onshore and determine when these boulders are moving. For my doctoral research, there's methodological improvements that were contributed to this field. So that includes movement modes, being able to distill from accelerometers, and all these other methods that we piloted and found to be useful. We found that boulders are moving during high tides, when they coincide with storms, when boulders are partially submerged, and we have much larger apparent Froude numbers than typically found in storm event literature associated with this field. And continuing work will work on offshore wave characteristics. And I know my time is up, so I'm just going to provide one brief slide on broader impacts, where I believe it's important to not just conduct the science, but also to be able to communicate it and to use it in your teaching endeavors. And so to this effect, I've worked on drafting 2 virtual field trips. One is to Ireland, one is to a dam failure site, where we use field data and we're able to drop students in there, transport them to Ireland, and they can look at boulder transport. And I've been privileged to teach a couple of field schools as well. As I look ahead, I'll continue working on coastal boulder deposits. I also plan to move from natural to engineered systems, so looking at dynamic revetments, cobble beaches, hard armoring, riprap on shores, and continue working with the numerical modeling as well and sediment transport. So thank you for your time.
So we have time for some questions. If you want to just raise your hand or drop your question in the chat. Go ahead, Lee.
Yeah, so thanks so much for the interesting talk. I really enjoyed it. It's very different than things that I usually see, and so it's very cool. I had maybe kind of a specific question about the iPhone LiDAR. I guess you can't get the bottom surface. And so I'm curious if you can comment, like I'm thinking that's probably a pretty small like potential impact on your volume measurements, but like what does that mean for your drag forces, your frictional forces, and some of those aspects?
Yeah, no, that's a great question. So in every case except one, we were not able to scan the bottom of the boulders. So we just used a simple polygon to close it. Even by doing so, our volume estimations were smaller than the cuboid estimation or when you apply a correction coefficient. And so we consider it to be better, but there might be other ways to close it, I guess, to answer your first question. The second question, so the drag area for these boulders, it's assumed to be the longest axes by the boulder height. So it's the projected area. So in that case, the bottom of the boulder does not play a role with respect to drag, but it would play a big role with respect to friction. And so, this model I would say is simple enough for application to boulders where we don't know what the waves looked like. But if we wanted to improve it, looking at that ground contact surface area definitely would be one area. And, is it touching the rock completely when it's moving? Is there air? Is it kind of this multi-fluid flow? There's so many questions that kind of get brought up when you think about that. But no, good question.
So just to clarify, so for the frictional forces now, like, do you just assume it's flat and just like it touches everywhere?
Yes. So you can set a slope. For ours, we assume a slope of 0, and then the frictional force is governed. It'll have the lift and drag components, and then we have the field set coefficients from the dynamometer measurements.
Awesome. Thanks again. Yeah. Super interesting. Really enjoyed it.
No, great question.
We have time for maybe one more quick question.
I can ask one more quick question.
Go ahead, Lee.
Are you able to differentiate like the floor movement from the boulder movement? Like I was just like some of your photos, it kind of seems like the floor is basically just rock, and so maybe not so much. But I'm just curious if it's like differential movement of boulders with respect to each other, or if potentially they're all moving together with the floor.
Yeah. With the floor, are you referring to
The ground.
the limestone platform? Yeah. So on the limestone, they will leave these abrasion trail scratches. So we'll be able to see their transport pathways. What we saw in the first 2 years is all the isolated boulders that were much smaller are moving in the same direction on these platforms back to the ridge, but the ridge was not adjusting. However, in the third year, there were much larger storms. And so boulders in the ridge were moving with respect to each other, which definitely makes interpreting those accelerometer orientation change results more tricky. So we find those are more noisy because they're constantly shifting in the ridge. And in the third year, some of the platform did get ripped up and created its own, created new boulders. So linking that to wave energy is also something I'm interested in.
Thank you.
If that answers your question.
It does, it does.
Awesome. Thank you.
Great. For the interest of time, thank you, Hannah, for your presentation. Thank you, Lee, for your questions. We'll move forward with the next speaker, which is Dr. Ke Zhang. Dr. Zhang is a postdoctoral researcher in the Department of Civil and Environmental Engineering at the University of Michigan. He earned his PhD in Energy, Environmental, and Chemical Engineering from Washington University in St. Louis. And he has expertise in both environmental engineering and biology. And his research focuses on environmental biochemistry, aiming to understand the biorisk posed by harmful organisms and biomolecules, and to engineer novel solutions for environmental and public health challenges. The floor is yours. Thank you.
Thank you thank you for introducing me. So I just want to briefly, wait a minute. Okay, so before I start, I just want to briefly mention that I have mixed background, education background. So for example, before my PhD, my education is totally in biology. And after I came to the United States, I found interest in environmental engineering. So my PhD and my current postdoc work is in environmental engineering. So my research will use both the method from environmental engineering and from biological science. So today, the topic is about the environmental biorisk assessment. And here I will use two examples to illustrate this topic, which is influenza virus and genetic modified crops. Environmental biorisk refer to potential risk and hazard posed to human and the environment by this harmful biological agent. So this harmful biological agent can be bacteria in the agricultural environment that will contaminate food and cause human disease. Or they can be respiratory virus such as SARS-CoV-2 or influenza virus that are transmitted in the environment. Or it can be the genetic modified crops that may pose hazard to this beneficial insect in agricultural environment. Or they can be antibiotic resistant gene that will result in serious infection and will also affect human health. So when you check this harmful biological region, you may notice that many of them are relevant to nucleic acid. It is not surprising that because nucleic acid play an essential role in living organisms and can be also found in dead organisms. So therefore, to understand environmental biorisk, it will be understand the nucleic acid in environment can be a very important part. And specific to myself, I'm particularly interested in RNA-associated environmental biorisk. That is because compared with DNA, RNA has broader relevance. For example, many viruses that affect human health, they are RNA viruses, such as influenza virus, SARS-CoV-2, norovirus, and many other viruses. And also, RNA is relevant to some emerging genetic modified crops. And more importantly, compared with DNA, so the immune fate of RNA is relatively less known. And this uncertainty will pose some challenge in the environmental biorisk assessment. Specifically, this research gap can be the environmental fate of RNA, or when we have RNA measurement in the environment, how we can interpret this data. And these two research gaps will be the two parts of my presentation today. So in the first part, I will use RNA molecule from genetically modified crops to illustrate the immune fate of RNA. And in the second part, I will use RNA signal from influenza A virus to provide insight into the interpretation of the event RNA signal. So for the first part, genetically modified crop. So a typical trait of genetically modified crop is their resistance to the pest. And this type of genetically modified crop is very common in the United States. So specifically, so scientists had engineered these crops and let them produce DSRI, which is double-stranded RNA peptide within crop tissue. And therefore, when pests consume on these crops, the DSRI peptide will enter the gut of these pests, and then they will trigger the RNA interference of the pest cell. So the result is that these pests will be killed. And this is known as the RNA interference. So these crops are also known as RNI crops, or these pests are known as RNI pests. So the core part here is the DSRA molecule act as peptide. This DSRA peptide from genetically modified crops has shown to be very effective to control the target pest. However, when harvest these crops, there will be some crop tissue live in the agriculture field. So this will also release some DSR peptide to the environment. So will this cause some risk to this non-target insect? For example, this butterfly, this bees, this beneficial insect, will they be affected by the DSR peptide from genetically modified crop? That's something we want to understand. And to understand that, we need to have ecological risk assessment. Ecological risk is relevant to both hazards and exposure. The hazard or the toxicity depends on the DSR sequence. And the exposure will depend on the immune fate of RNA. So currently, there are probably 30 or more than 30 types of DSR peptides, and they are different in their sequence. So when they have different sequence, they will have different toxicity. But even have different sequence, their environmental fate will be similar because they are all similar DSR molecules. So why say that exposure depends on their immune fate? That is because when DSR peptide decay fast in the soil, the environmental concentration will be low. However, if this DSR peptide does not decay and always persist in agricultural environment, so you can imagine that each year will grow a new batch of these crops. So the DSR peptide will kind of accumulate in the agricultural environment. So this will cause a relatively high environmental concentration. And therefore, understand the environmental fate of DSR is the key here. So to understand the environment of DSRI, there will be two specific questions. First question is that, is RNA chemically stable? And secondly, if RNA is stable, is DSRI or single-strand RNA has similar stability? So here I want to mention that DSRI or double-strand RNA, it is annealed by two single-strand RNA. So therefore, single-strand RNA and double-strand RNA, they have similar primary structure, but the only difference that double-strand RNA is annealing of two single-strand RNA molecules. So they share very similar structure, but just different in the secondary structure. The first question, is RNA chemically stable? Single-strand RNA is known to have alkaline hydrolysis, that its hydrolysis can be catalyzed by hydroxide in aqueous solution. However, currently, it's relatively unknown about the alkaline hydrolysis of RNA at neutral pH. So many biologists will know that the half-life of messenger RNA in cell is approximately just several minutes, and therefore, a representative opinion is that RNA is considered as very unstable molecular, and no matter it has single-strand structure or double-strand structure. So to test this research question, we synthesize several RNA molecules, including single-strand RNA and double-strand RNA, some long RNA molecule and some short RNA molecule. And after synthesizing RNA molecule, we incubate in environmental relevant condition, that is pH 7 and ambient room temperature. So here, because I want to check the chemical stability of RNA, so I want to exclude biofactor. Therefore, I use the RNA-free protocol. I have to use RNA-free protocol because RNA is very common in environment. For example, it can be present in the soil that have microbes, or that are human. We also produce RNAs on our skin, so we should be very careful when we do this experiment. So here is the result. I synthesized 4 RNA molecules, including 2 short RNA, 2 long RNA, 2 single-strand RNA, and 2 double-strand RNA. So you can see that all these 4 RNA molecules, they remain stable at neutral pH up to 74 days. And you may also notice that I actually use A replicate for each molecule, and none of them show degradation. That indicates that RNA, no matter it's single-strand in the form of single-strand RNA or in the form of double-strand RNA, it is always very stable, can be stable up to several months. So then the next question is, do DSRI and SSRI exhibit the same alkaline hydrolysis rate? So as I mentioned, single-strand RNA and double-strand RNA, they share similar primary structure. They only differ in their secondary structure. And my hypothesis here that because the double-strand RNA has this double helix structure, and this double helix structure will block the intramolecular attack. So therefore, I hypothesize that DSRI will be more stable because it have less likely to have the phosphorodiester bond breakage. So to test this research question, again, I synthesize both single-strand RNA and double-strand RNA. And here I use alkaline condition instead of neutral pH condition. That is because I already test the stability of single-strand RNA, double-strand RNA, and neutral pH. So they decay. So they are very stable. So I was not able to measure their degradation rate. So I had to increase the pH to be able to measure their degradation. Another reason that because the inside gut sometimes have a extreme alkaline pH. So if I use alkaline pH, it will mimic the inside gut. So therefore, if there is degradation of RNA, at this extreme pH, that means when the insect consume this DSI molecule, they can degrade that. Otherwise, so they will be persistent in the insect gut. So therefore, here I synthesize two RNA molecules, single-strand RNA, double-strand RNA. You can see that I incubate for more than 200 minutes. So I only see the degradation of single-strand RNA, but double-strand RNA do not, their concentration do not change much. Therefore, this means DSI exhibited much lower alkaline hydrolysis than single-strand RNA. So I know most of you may be less familiar with DSI from genetic modified crop, but I want to mention that our study have broader implication. That is because even for single-strand RNA, the self-folding is very common. So once the single-strand RNA fold by itself, it will have some double-strand, so DSI region. So even if it is single-strand RNA by itself, some region will be similar to double-strand RNA. And you can imagine that this double-strand RNA region will be more stable than the non-folding region. So as an example, here is the genome from MS2. MS2 is a common ssRNA viral surrogate. So I predict the self-folding of things of this genome, and you can see that the self-folding is very common. So as a summary here, I see both ssRNA, double-strand have no degradation at neutral pH, and at alkaline pH condition, I saw that DSI is more stable than single-strand RNA. I want to mention that ecological risk of DSI peptides will consider many complex environmental conditions. Although here I show that DSI is more stable than single-strand RNA and has no degradation at neutral pH, we cannot just make conclusion that DSI can persist in environment. That is because if you use different environment, it may have different, so DSI may have different stability. For example, in my private study, I show that when this DSI go into the soils, some mineral in the soil may catalyze the hydrolysis of RNA by surface catalysis. So then let's go to the next part, RNA signal from influenza A virus. Influenza is very common in the United States and worldwide, and it's mainly transmitted in indoor environment. So therefore, we can use indoor surveillance to track the respiratory virus and to help us to understand the community health. And specifically, we can measure the respiratory virus in air on surfaces or in air conditioning filter. So after getting a sample, we can either measure their infectivity or measure the viral RNA concentration. However, in real field measurement, we often measure the RNA concentration instead of infectivity. That is because RNA measurement is much more sensitive and faster than the infectivity measurement. However, this will also cause some problem. For example, so when we measure the respiratory virus concentration in the indoor environment, we have positive detection. So the question will be, where this RNA from variable or dead virus? Or when we have negative RNA detection in the indoor environment, so there will be questions that where this negative detection is caused by the absence, so there is no presence of virus, or there is presence of virus, but the viral RNA just decays so fast, so we cannot measure them. So therefore, although we can easily get the RNA measurement data, it's hard to interpret this data. So that's my goal, to better interpret the data. Here is the experimental setup. So I use 3 viruses, influenza A virus, and also another 2 surrogate viruses. So I mix them with saliva that kind of mimic the virus from human mouth. And then I deposit the mixture onto surfaces and incubate them in metal chamber. This metal chamber can mimic indoor environment such as temperature and humidity. So after incubation, I will measure their infectivity and RNA concentration. So here, I measure both their infectivity and RNA concentration. You can see that for the 3 viruses I measure here, so the left part is the infectivity. The infectivity will have decay within several days. However, when you check the right part, RNA measurement, RNA concentration, that I incubate for 2 months and they had no much degradation. So the RNA is much more stable than the infectivity. So just now I used 23% of relative humidity. So this is just this is the one humidity condition. So previous study indicates that if I use different humidity condition, I may have different infectivity decay kinetics. For example, here I use 3 humidity condition. The low, so relatively dry humidity condition, medium humidity condition, and humid humidity condition. So you can see that, so for example, this square is the phase 6. You can see that this phase 6 decay most rapidly at this neutral humidity condition because it reached the detection limit much faster than low and more humid condition. So this is consistent with the previous study. And if the humidity can have an effect on the infectivity, will it affect the RNA persistence? We also check that. You can see here that, so the left part is the infectivity and the right part is RNA measurement. You can see no matter what humidity condition we use here, RNA, they are always stable. So they always decay much slower than infectivity. And therefore, you can imagine that if we use, so if in the indoor surveillance, we use RNA measurement data to estimate the transmission rate, we will greatly overestimate transmission rate because even long after this virus lost their infectivity, their RNA signal can be always persistent there. So in the figure, you do notice that the RNA concentration didn't change much over 2 months of incubation. However, this part is a little bit inconsistent with previous study. So for example, here is a review paper. It reviews some like high-risk area. For example, there are all hospital rooms that host the patient. However, in this high-risk area, the virus RNA detection rate is often very low. So that'll be interesting because if in our study, we see the RNA never degrade over several months. So that means it should always accumulate and no degradation. So why in field measurement, they often see low detection rate. So to explain this, our hypothesis is that the RNA may have a cell degradation when there is very humid condition. That is because also in our 2 months incubation, we use fixed RH condition. In real indoor environment, the humidity will change, for example, with the temperature or with the air condition operation or with human activity, such as breathe or when you take shower. So therefore, we compare the fluctuating RH condition between low and high humidity compared with fixed RH condition. You can see that only the very RH condition can cause accelerated RNA degradation. And in all other fixed humidity conditions, there are no RNA decay. So therefore, we prove our hypothesis that RNA do have accelerated degradation when there is very humidity. And we propose a mechanism for the accelerated RNA degradation. Specifically, we consider there will be non-structural protein and structural protein of virus. So the non-structural protein will relevant to infection, and the structural protein will not relevant to the infection. At phase arch human condition, so it lost infectivity, but the structural protein keep intact. This will protect the viral RNA. However, when there is very arch condition, this will damage both protein, and this will expose the genome to the RNAs in the saliva, and this will cause RNA degradation. So the arch fluctuations that here will cause hydration and dehydration of the protein. This will be similar like the freeze-thaw cycle on the viral RNA. So they may have similar mechanism. So as a summary here, we found viral RNA is much more persistent than their infectivity. And we also found that viral RNA has accelerated degradation when there is varied humidity. And finally, I want to thank my PhD advisor, Kim Parker from Washington University, and my current advisor, Krista Wigginton from University of Michigan. So that's all.
Thank you, Kay. Thank you for your presentation. Does anybody have any questions? Nicole, looks like she has a question.
Yeah. Hey, Ka, thank you. That was great. My question is, of course, about the flu part, or the RNA, and the alternating humidities. It makes complete sense. Like you say, we have difficulty detecting bioaerosols or RNA in places where people are shedding. Are there, like, even on the order of like minutes after we know someone's shedding virus, we still can't detect it? Do you think that this mechanism is happening that quick, or are there other alternative hypotheses for why we just don't detect anything in those cases? Like maybe they're just not, I don't know, not expelling as much into the air as what they're shedding maybe in their nose.
So you mean the infectivity part, right?
No, even the RNA, like it in those studies you showed like pretty low percentages, and some of them are measuring in spaces where people are actively shedding. So like a virus might not be sitting on a surface for days and going through humidity. So what do you think might be happening there when maybe it's on shorter timescales?
Yeah, I think one change in aerosol studies that the sample amount is so low compared with sample in aqueous solution. So I think that's because the initial amount will be so low, even there are no degradation, the initial amount will be very low. So like sometimes often close to the detection limit. So therefore, if the concentration is a little bit lower than that limit, you may have negative detection. That's my understanding.
Okay, so detection limit's not necessarily rapid degradation.
Yeah.
Thanks.
Lee, go ahead.
Yeah, thanks, Fab. Thanks for the nice talk. Very interesting. Again, a little bit outside of my area, so hopefully my question makes sense. But for the first part of your talk, and kind of looking at the stability of the double-stranded RNA, I'm curious if you've kind of taken that to like implications for the amount of double-stranded RNA that should be applied, as pesticides or the frequency of application. Like, can you use your results to inform like farmers on how much and how frequently they should be applying these pesticides, to avoid overdosing or underdosing or anything like that?
I think it is possible if we get more data. So I mean, just from my study, probably like as I mentioned here, ecological risk assessment, like you should consider a lot of environment. So let's say, for example, if we consider temperature, if in one area has relatively higher temperature, the RNA decay so relatively faster. If in a cold area, may kind of RNA persist for a longer time. So like in aqueous solution in surface water or in soil. So this will involve many like complex environmental conditions. Like I may just study several relevant conditions, but there'll be still some uncertainty in other conditions. So considering this, I should say like maybe like after years, probably when there are more data, when we have more data, it will be possible to predict. But currently, we probably is not that confident to see that.
Makes sense. Oh, thank you.
Thank you, Kay. I think I'll let Jessica wrap up. I think we're almost at time. So.
Yeah, thank you so much. We're right at 1 p.m. So let's just thank both of our speakers again, Hannah and Kay, for excellent seminars. We have 3 more seminars coming up next Friday, which is the 14th is our next seminar. And then we have 2 additional seminars on the 21st and then December 5th. So we'll be posting about those to the AEESP listserv. So make sure you keep an eye out for that. And this is the same Google Meet link that will be used for all of the seminars. So again, thank you to our speakers. Thank you to the committee for your help in organizing this. And enjoy the rest of your Friday.
All right, thanks.
Thank you all.